Thank you very much for inviting me to this very wonderful meeting thus far. Again, I'll start slow because probably some folks will come in hopefully during the lunchtime. But here is sort of the bottom line of what I want to talk about in this very first slide. We all know a lot about long-term caloric restriction, which does a lot of things to these animals. You start caloric restriction at a young age, 5 months of age. You keep their weight controlled. They're actually a little more active. And we and many others have extensively studied the beneficial effects on the heart, the muscle, peripheral nerves, and a variety of other tissues. So again, this long-term caloric restriction is very helpful and beneficial to these animals. There are many mechanisms that are turned on with caloric restriction. The one we'll focus on today is autophagy. I think it's one area we can intervene. There are some nutritional compounds. There was a question on this earlier that can activate autophagy. And I'll describe how autophagy works, how we can pre-activate autophagy, and then induce a free radical stress, and we can see cell protection. That's all great and nice to do this in a cell line, but at the end, somebody's talking there. It's bothering me. Sorry. The harder thing we'll try to do is take older animals and caloric restrict those for a period of about 2 months, and we're trying to see if autophagy is upregulated, and then give them a stress. So that's more challenging, old animals to intervene and to cause cardioprotection. The final point I'll make is that interventions that use multiple treatments are the way to go for the elderly adults that are becoming aged. And we're going to have to think cleverly in combining, for example, exercise, moderate caloric restriction with nutrients or drugs to get the full effect. That's the final bottom line I'll make in this talk. Aging is complex. You can see many major pathways or processes that occurs in the cell. You know, probably genome instability on the top is the most relevant one, but you can see many other processes that occur. Inflammation, stress resistance, we'll talk a little bit about that. Protein synthesis, quality control, but you also see autophagy at the end. Again, all these processes take place, and by no means, you know, can a single compound do this and try to block all these pathways of aging. Tilman Groen had an excellent presentation on aggregated proteins, so I'll give you the punch line. If you have aggregated proteins that are smaller in size, they can be removed by the UPS system, the ubiquitin system after ubiquination by the proteasome. If aggregates become too large or mitochondria are becoming damaged, you need macroautophagy. So if the products are too large, you need to remove them by autophagy. So the autophagy process is multifold, and there is a variety of proteins involved that create this sac, kind of this garbage bag in the cell to take these larger molecules or aggregates or damaged mitochondria and remove them and carry them to the lysosome. There's lysosomal degradation. They fuse with the lysosome, and then the organelles are recycled, and they can be reused in the cell. So it's a self-eating process, and it's very well regulated. And I'll also demonstrate to you in this talk that mitochondria that are more damaged are getting turned over, which is the crux of the issue. You want to take away these older mitochondria or these damaged mitochondria to prevent extensive oxidative stress. This is probably the most powerful slide and paper, and I'll just give you an example of homeostasis in old animals. The entire talk will be focused on cardiomyocytes, but this is just one slide on hepatocytes. And the bottom line is, it doesn't show real well, but if you're young, you still see red here after ischemia reperfusion injury, a severe injury in these liver cells. So there's an extensive amount of cell survival. We've all documented this in this paper. But in older animals, the cells die. So if you pre-treat these animals with viruses, meaning viruses where you can overexpress some of these autophagy proteins, like ATG4B or Beclin, they're part of the sac forming. So they're the proteins that are actually lacking at old age. You can entirely prevent cell death. This is ischemia reperfusion. A lot of cells die. These are pre-treated with these adenoviruses to overexpress those proteins. Again, I won't have time to present this paper, but if you have time in your daily lives, which is very busy, read this paper. It's a very, very nice paper. So today I'll focus, the first project's on cell culture, cardiomyocytes. And in vitro, we'll try to enhance autophagy, and we wonder if they prevents oxidative stress injury. Are these cells surviving? Are they respiring better? And then we'll go to the more challenging jump, where we take older animals and we pre-treat them with pro-autophagy compounds. So here's the experimental design. These are the in vitro models. There are 2 cell lines, one derived from a mouse, one derived from a human. One is atrial, one is ventricular derived. The drugs we typically use are antimyosin A or doxorubicin. Antimyosin A produces free radicals in complex 3, doxorubicin mainly in complex 1. And then some of the autophagy inducers that we'll use are rapamycin and resveratrol. And I'll show you another list of some other compounds that we're testing. But you have a pre-treatment. Of course, it's important to upregulate autophagy, and hopefully you sustain autophagy for a prolonged period of time, so there's protection. You have your oxidative stress with AMA or DOX, and then you wait a couple hours, you wash the media, and you add your pro-autophagy compound again, and then during different time periods, you do your biochemical analyses to see if cells survive, if they're respiring, etc. Is autophagy upregulated? Can I block autophagy? And so on. So this is just, the first one is very simple in the sense that AMA is used. It produces superoxide radicals in complex 3. It's hard to see, but the bottom line is there's a dose-dependent effect of AMA on the amount of free radical production. There's also dose-dependent effect on the membrane potential. There's a decline in membrane potential as you increase the dose of the drug. There's also oxidized DNA damage, and RNA damage is also increased. And you can see here that the cells that are viable with the higher dose is the lowest levels of cells are surviving. So we're testing just simply here, is the drug working, and what's the cytoprotective, how well does it kill the cell? So how to measure autophagy is not easy, but several techniques have helped us identify autophagy, not only by Western blot analyses. But one of the techniques is used here, and you can see small dots forming. You can add this gene fluorescent protein. It can be fused to one of the key autophagy proteins, LC3. So you can have this marker, this GFP LC3, and you can now count dots or accumulations of these GFP LC3s, and then you can count them and say this is how much autophagy is going on. So this is a powerful way to determine autophagy. The other thing that needs to be done is, because autophagy is a constant process of forming these bags which has some of these proteins in it, and the degradation steps, we need to add inhibitors, like beflamycin A1. You do this about 4 hours before you terminate your experiment, so you accumulate the LC3 GFP labeled protein, so you can easily quantify it. Again, so there's some technical issues to help demonstrate autophagy. So here's, in this case, it's a compound called rapamycin. We're all familiar with that. And we show that it induces autophagy in the HCl1 cell line. And to go over some of the key slides is, sure, you can see the LC2, LC1 increase in its ratio. And again, you need to do this with the beflamycin A1. You can also look at the number of puncta, the green dots, and you can count them, and you can see that autophagy is upregulated due to the pretreatment of rapamycin, typically 24 hours or 16 hours prior to your experiments. Also, you can use multiphoton imaging, and you can demonstrate mitophagy. In red, it shows mitochondria, and if you treat the cells with rapamycin, you see an overlap between red and green. Again, green is the autophagy or mitophagy process, and you see a small yellow puncta appear as well. So again, yellow or green and red makes yellow, so you can demonstrate that mitophagy is active. Well, that's all great, but the key is to determine, are these cells protected by rapamycin? And that's demonstrated in this slide, where you can see your control cells are all healthy at 100%. Here's your rapamycin. It's not causing any negative effects. And then here you have your oxidative stress inducer, and you see a marked drop in cell viability. And this is rescued by pretreatment of rapamycin. And again, this is done in both cell lines, the HL1 and the AC16. I mean, we submitted the paper, they loved the paper, and then they said, repeat everything on another cell line. So that's why we are using 2 cell lines. And then it got accepted in autophagy. We were very happy with that. So again, here we have more cells surviving. Well, that's a big deal. But I think what's even more important is this experiment, where we show that these cells are actually respiring. Are the mitochondria doing better in those cells? Yeah, and we did this with high resolution respirometry. We did not have to permeabilize the cells. We could just add them to the chamber and look at the oxygen consumption. And again, the story is pretty simple. Control RAPA, no change. Here's your drug. And then again, the pretreatment rescues, not entirely. I mean, I think there's few drugs that can entirely blunt the insult, but there's a significant attenuation of the amount of damage. And some additional images were made to look at the morphology of these mitochondria. Well, that's great, but the next question is, does it work through autophagy? Is autophagy one of the major protective mechanisms of these types of experiments? And the answer is yes, to a large extent. So what we did here in simple terms, and then we'll look at the data, is we used pharmacological ways to inhibit autophagy, or genetic tools like shRNA to inhibit specific steps in the autophagy process, like blocking Backlin-1. So it can't have an effective autophagy process. And both those experiments were successful. You can see here, looking at cell viability, these are sort of all your control experiments. And here's where the action happens. You add entomycin A, you see a decline in cell viability, rapamycin pretreatment rescues it, and then if you knock out, in this case pharmacologically, you inhibit autophagy, you're back to the AMA alone experiment. Here's the genetic experiment with the genetic tools, and you can see the same effect. Here, control experiments. This is a control scrambled. It's also an entire control experiment to show there's no effect of a scrambled shRNA that's added. And again, here you can see a marked decline, and in the scrambled form, you see, again, it's blunted. So these mechanisms are really, to a large extent, through autophagy. So there was a question earlier this morning, I think related to some products that can turn over proteins better that are damaged. And to start at the bottom, these are not secret compounds. They're all available in the literature. But we and others have some non-FDA approved drugs that in a variety of cell lines, and here's the first list. These are the activators. These are the sources where they come from. And then these are the targets and how they work in the autophagy process. But we're testing some of these non-FDA approved compounds and see where they act in the autophagy process. Of course, there's FDA approved compounds, and a lot of these are either used for cancer therapies or some of those for a variety of cardiovascular abnormalities. And then there's, of course, at the top of the list, there are natural compounds that can upregulate autophagy. And most of the studies done to date, they've only looked at some of the proteins that are increasing, but they haven't looked at the entire autophagy flux in these cells. Again, adding the inhibitors, the more difficult experiments that are to be done. So we're testing these compounds. Some are very straightforward: vitamin D, curcumin, caffeine, in their ability to upregulate autophagy in these cell lines. Okay, the second part is a little more interesting because we're starting here with a Fisher-Brown Norway rat at an age of 24 to 26. There was a question earlier, okay, how old is this? Well, in this specific strain, every age is very specific for a strain. In this specific strain, if you want to translate it to humans, it's about 60, 70 years of age. You can get to this number because you know approximately these animals, they die at age 32 to 36, give or take. So that's kind of their maximum lifespan. This is also an interesting age to study because cardiovascular issues are showing up in this animal model. They have basically diastolic function, left ventricular failure, and it's a nice time potentially to intervene. But it's also a challenging time. Is it too late or is it not? But again, we're starting at 24 because there's not too many serious events ongoing, but they really are detectable at 27. So we have a short intervention period, and after that, the 2-month intervention, we add a drug. In this case, it's doxorubicin that causes free radical production in complex 1. Resveratrol is used in this study, and no need to tell you about resveratrol. Some of us had some resveratrol for lunch. I'll wait till a little later. But we're very familiar with the French paradox and the potential of resveratrol, which many now show it's not necessarily an antioxidant. It does many things at the gene level, so it's a gene regulatory compound, and we'll also see that it can upregulate autophagy. So we're going to give these animals resveratrol, but like I hinted to you earlier, we're also are going to combine this with a moderate amount of caloric restriction. 20% is well tolerated at an old age in these animals. Again, people are always so worried about caloric restriction and think this is very hard to do in humans or in animals, but it's just trying to maintain normal body weight and it's giving a small hormetic stress during their life or later in life to these animals. So some of the groups will also have caloric restriction plus resveratrol. And what we'll see is that that's where we see the effects. That's where we see cardioprotection. So here you have your acclimation phase. You start your intervention in these old animals, 20% CR. I should put a glass of wine here instead of these grapes, although we're not giving them wine. The resveratrol is fed in their diet, and it's actually flavored with bacon to make sure they eat it all. It's a little trick that will be explained in the paper to make sure they eat their entire palate. There's an intervention period, and then you add DOX. And again, a quick note, DOX is one of the most common chemotherapy drugs used in humans and animals to treat cancers. And it has a side effect, which is it damages the heart. So what are we looking for? Whether pre-treatment with caloric restriction by itself or in combination with a low or a high dose of resveratrol can induce autophagy in the hearts of the 24 to 26-month-old animals. And if they can protect the heart from against oxidative stress while upregulating autophagy. So this is just, in brief, the design. You have your ad-lib fed animals. Ad-lib fed animals with resveratrol, 5 milligrams per kg, or 50 milligram, the higher dose. And here are the very interesting groups. This is CR combined with the low and the high dose. So in the initial studies, in the initial pilot studies, using slightly smaller numbers of animals, we will publish this, we found very little changes in autophagy induction in the ad-lib fed animals. Like I indicated to you, you really need to stimulate these animals by multiple ways to induce autophagy. So the data that I'll show you are on the ad-lib, the 20% CR, and the 20% CR, and the 5 and 50 milligram addition per day. So autophagy measures in vivo are more complex. And some of these markers actually were also used in the cell culture. But what we can do, for example, is follow the levels of P62. And P62 is involved in the ubiquination process, and it's also degraded by the lysosomes. So as this marker disappears, you can give it gives an index of the levels of autophagy. That's just one little biochemical slide here to show you. So if we go in this example from right to left, and I know the changes you can see are very small, but again, envision old animals, only a 2 months intervention, but you're going to see they're cardioprotective. You can see a small but significant change in Beclin-1, one of the important autophagy proteins. You can see a disappearance of the P62. But as you can see here, this LC2-LC1 ratio, this is when it converts from LC1 to LC2. It gives an index of autophagy. You can see there's not too much change. In the cell experiments, to go back to that, you can use inhibitors and have an accumulation of this LC2-to-1 ratio. You can and you cannot do this in animals. You can in the sense we did not do it. You can inject chloroquine and do this in additional experiments. We may have to do this in the future. But nevertheless, it's suggestive that autophagy is increased. But we need to use some inhibitors and inject some inhibitors in the heart to really show an accumulation of these damaged mitochondria in these vacuoles. So we looked at cardioprotection, and we looked at apoptosis and creatine kinase. We're all familiar with apoptosis, and creatine kinase is a major muscle enzyme, typically used even in clinical settings. And you can see again the lowest levels of apoptosis or creatine kinase activity were detected in the caloric-restricted animals plus the resveratrol-treated high dose. And again, very, very interesting, and we're doing some more experiments to show the amount of damage that was attenuated. This is a published paper a while back where we showed that long-term, 10%, I went back to this paper and I looked at it, and we did 10% caloric restriction plus resveratrol for a 6 to 8 month period of time. So we started at about 15 months of age, waited till 25 months, and what we did, we showed cardioprotection in the sense we showed better functioning of the heart. So there's actually better contractile properties in the heart, especially in the animals that were caloric restricted and received resveratrol. In this case, it was a lower dose, but for a longer period of time. So in summary of this second part is CR plus resveratrol decreased the P62 abundance, increased Becklin-1 in the left ventricle homogenate. Doxorubicin enhanced apoptosis, and this was the most effective treatment to attenuate this. Also, CK was attenuated. We did some additional analyses. LDH is not very specific, could also be liver related, but it was also attenuated in this animal model. And then importantly, CR dilate alone or resveratrol alone, but the combination was the key. Oh, we had some body composition changes I didn't show. I should put that in there as well. But we had some loss in fat mass, and we had some preservation of lean mass during this very short period of time. So humans, I'll show 2 slides and I'll be done. Can we do some of this in humans? And I certainly believe it's very important to test some of this in small pilot studies and isolate muscle samples. So we did muscle biopsies in older, overweight women. And we had an exercise program combined with a moderate weight loss program. Effect of a weight loss plus exercise program, and we looked at the overall quality of the muscle. I'll just jump quickly to the data, but in the few proteins we analyzed, again, we don't have a lot of muscle, we could find upregulation of some of the autophagy proteins in the intervention groups compared to the control. Here we also see proteins important for ubiquination and turnover of potentially damaged proteins. And again, you may wonder what happens to important mitochondrial proteins like PGC1 alpha, TFAM, and you can see either the gene expression or protein levels were increased. So you still have, in these older obese women, you still have happy mitochondria. They're not getting all removed. There's actually a slight induction during this intervention time. So the punchline is that as we get older, we need to, of course, we need to move, we need to be moderately caloric restricted, but don't think you have to starve yourself. People are so worried about this caloric restriction work. You just need to have metabolic balance, I call it, during a daily setting. Because we know as we get older, we have ischemic stresses, we have a variety of sometimes psychological stresses, sometimes too much exercise stresses, and we need to pre-protect ourselves by taking some of these potential important compounds because we know during those stresses, especially when we get old, we have a heightened level of the amounts of damaged mitochondria. They could be very transient. You could be shoveling snow up north. You have an ischemic event. If you're ready for it, the fire truck is nearby, so to speak, and you can clear out those mitochondria very, very quickly. The chance of survival of individual cells is greatly improved. And again, we certainly showed this in the cell lines. And in the animal model, again, more experiments will need to be conducted. Here kind of also sums it up that, and this is maybe something I should have probably mentioned a little earlier, is that we need these combinational interventions. Caloric intake or exercise should have been put here. You can have mTOR-dependent compounds, or not only mTOR-dependent compounds like resveratrol. They work in multiple actions, and they activate a variety of proteins or inhibit them to turn on very effective autophagy. Again, not only one of these by itself is typically as effective as a combination, and we're looking into that as well. So I want to thank some of the folks that have done the work, Priya, Jinze, and the American Heart, National Institute on Health, and then our IDP program that funds graduate students. Thank you.
Questions from the audience? Who will you first?
I realize that this could seem innate question, but I just try to draw some conclusion for our life. And what I learned, sorry, John, that we have, in order to protect our heart, we have to have a caloric restriction 20% and drinking, if calculated correctly, 265 liters of wine a day. Don't think it doesn't fit too much to each other. Well, it was a joke.
I know.
I don't know the answer, but...
Well, many studies, including ours, show that actually very low dose of resveratrol works quite well. A public statement by some famous scientists when they say, and I just met Sinclair this weekend again, if they say you need to drink 100 bottles of wine to get the same effect, 200 bottles, the data shows you can do it at lower levels. So if science is important, they're making their own individual compounds to sell these, so there's definitely a conflict of interest. So low levels of these compounds, actually, they work. 5 to 50 milligrams work in inducing autophagy.
And from that, I calculate that.
Yeah.
Kilogram per kilo.
But besides wine, we can get it through peanuts, nuts, there's grapes. Wine is more fun, but there's a variety of things we can do during our daily life, not only to intake more resveratrol. I indicated in the list I had, a very selective list, about 10 other natural compounds that can increase or get you ready for the autophagy process.
Very nice there. I just have a couple of comments and maybe a question for you. So you know that resveratrol doesn't really induce canonical autophagy. It's a bacterium-independent system. That's why probably you don't see much upregulation of FN1. And as far as your P62 is concerned, I mean, you have to be a little bit careful because P62 is under the transcriptional control. So now it's been shown that degradation of P62 may actually occur at the transcription level. So it may not be actually degradation of lysosomes. So you show here that P62
Good points.
settings or in vivo settings is your only measure to indicate autophagy, and your other markers are pretty dim. I suggest you should just do a simple electron microscopy to look for autophagy, because that would really rule out all these.
Or some EM, we're thinking some EM.
Because it's going to be difficult to inject chloroquine into these animals. They won't like it.
I think so too, yeah. I mentioned it, but it's a dirty experiment because it causes many toxic effects. So excellent comment. Again, like I indicated, we need to do more analyses to demonstrate autophagy, and specifically mitophagy is a little more challenged, but we'll do it with EM analyses.
Yes, please.
Very interesting work. I just have two questions. One that you related to your in vitro study. When you look at the cells and you pre-treated by rapamycin, right? And then you challenge the cells, you can see the protective effects. Have you tried, challenge the cells first to see whether rapamycin can rescue the damage? That's one. Number two.
I can answer that one quickly. No, we haven't done that. We will need to do it. I expect it's hopeless, but I think we need to do that experiment.
Number two, related to your in vivo study. When the animals, pre-treated with, actually the animals, when you challenge them, see, I'm sorry, the animals treated by both CR and
Doxorubicin.
the resveratrol. Can you see the extent of the lifespan because of cardio condition improvement?
That's a different experiment. And so basically you would injure the animals and then you would wait another 6 months and see how quick they die. It's a very costly, I wish there was NIH fund, it would be a good experiment, excellent experiment, be a very costly experiment to do, but I like the idea. You need to look at long-term outcomes of these animals. So what, short term, you have some cardioprotection, what about long-term after that? Excellent question.
Yeah, because you have a certain advantage because these animals are old animals, right?
Yeah,
Okay, thank you.
yeah, but old is important.
Christiaan, a question about the last slides. The one that you did with obese women, the question is first whether were African American, white or Latino, because the genetic makeup is important in terms of what is type of intervention. Previous to that, you show another slide where caloric restriction modulated the level of glucose and IGF-1 in plasma. Yeah, that was in rats.
Yes, yeah, that was mice actually, the long-term one was
The
mice.
long-term and in rats. And again, that was not a combination of caloric restriction and resveratrol.
It was 10% caloric restriction because,
No resveratrol there.
and resveratrol, so the combined effect. Yeah, so all groups were there. There was an ad-lib fed animal, a young group. You want me to go back to that
No,
day?
it's okay, it's okay. Because what I've seen is that the levels of IGF-1 were very decreased in the caloric-restricted animals, but no effect those treated with resveratrol. It was alone or in combination?
It was in combination, yeah.
And
The first part, I think it's very important to look at, you know, when you do a larger clinical trial, typically you have to include certain numbers of minorities, which is very important. That study actually had a lot of minorities. It was mainly African American women. It was a small pilot study. There were some Caucasians in there. But again, you know, the IRB requires you, and most studies require you to have a very homogeneous group in a larger clinical trial to represent all the...
minorities, but actually the clusters, because they will be amenable to one treatment or another. It's not the same. I mean,
So
we are
I agree. So secondary data analyses on a larger trial will need to tease out your question. Is there difference response? Are there responders or non-responders? But this is an excellent point because there's a lot of literature out there now in preclinical studies that resveratrol alone doesn't work as well, but if it's combined with exercise. So some people are responders, but some need a double treatment. But that's because of genetic differences. So we're getting smarter as we move forward in that sense.
Thank you very much.